Good morning, good afternoon, and good evening, wherever you are on the globe today. My name is Joe, I'm your host, and I want to thank you all for taking time out for what will be another very informative presentation from the experts at Specs. So, welcome to our presentation, Amplify Your PCR Success. And this webinar is part of the Science of Real Life webinar series, and we run these pretty much every 3 weeks, so keep your eyes out for these moving forward. And before I hand over to the amazing presenters today, I'm just going to go over a little bit of housekeeping and get you guys settled in. First of all, though, something significant has changed. Our business has moved from being known as Cole Palmer to now being called Antilia Scientific. And you can find out more about the change and why we changed at antilia.com. But don't worry about going to antilia.com. I'll put the link in the resource box for you all, so you can visit that in your own time. Another thing about this experience today on the webinar, you can actually rearrange all the slides and resize them just to make it personal to you. Some of you have larger monitors, some of you are watching on mobile phones. So please feel free to maneuver the objects on the screen to suit your needs. And most importantly for now is the Q&A box. So as we're presenting today, you can all ask questions. And what we do is at the end of the presentation, we have a dedicated section for our Q&A. And we read the questions out, and we do get a lot of questions, so please forgive us if we don't read out all of your questions. But what we do is we set up a Q&A document. We list out all the questions that are asked at the webinar today, and our experts will answer those questions, and we email it out to all the attendees. So please do ask questions, and just bear with us if we don't get to them today, but we will address all the questions in our follow-up Q&A document. And finally, we have a short survey. We really appreciate everyone just taking 30 seconds just to have a look at the survey and answer as many questions as you can. It helps us help you, and it develops our service capability. So we're all about keeping our customers happy, and that's what we're trying to do today. So I'm just going to quickly introduce you to the panel. We've got Patricia Atkins, our Senior Application Scientist. You're going to hear from Patricia first. She'll start presenting shortly. Then we've got Alan H. Katz. He's our Director and Global Product Manager of Chemicals. Eric Smith is our Application Scientist. We'll see him in the lab a little bit later. Leo Anderson-Smith is our Director of Engineering in the R&D section. And then we've got Maddie from our EMEA Product Sales team. She's just here today for the Q&A part to add a bit of extra PCR expertise. So without further ado, I'm now going to hand you to Patricia to get us started.
Thanks, Joe. Well, like Joe said, good morning, good evening, or good night, depending on where you are. Today, we are going to talk about PCR and give you some insight on how to amplify your PCR success. But we're going to start out first by just covering some of the basics. And I'm sure that some of you know this like the back of your hand. Maybe some of you are new to PCR and are just trying to get your feet wet for a little bit. But just to make sure we're all kind of starting off with the same language, we're going to talk about the chemistry of DNA. So DNA is the master code. It is the chemistry of life, as they like to call it. And it's composed of some important components, the backbone of which is the phosphate and the deoxyribose sugar. So the phosphate and the deoxyribose sugar themselves are considered to be the nucleotide. So the sugar itself plus an oxygen is the nucleoside, and with the phosphate is the nucleotide. And I've created that cute little icon there with the circles and the five-sided pentagon there. And I did that for a reason, because if you try to stack these up in the way that they connect, you cannot create a straight line with them. It actually forces a twist or a curve. So that's why you kind of have that curving, that helix curving ladder effect on DNA. It's because the phosphate and the sugar, when they bond together, and then they bond to the base pairs, which we're going to see in a second, they force this kind of structure. So that gives us that characteristic twist. Now, the master code is essentially 4 compounds: the purines, which are adenine and guanine, and the pyrimidines, which are thiamine and cytosine. And they lock together like key and lock. So you have the pairing of adenine with thymine, you have the cytosine pairing with guanine, and you can see where the different components, where the different molecules bond together with those dotted lines. That's how they come together. And you don't have crossover. They are exclusive pairs for the DNA. And I like to remember the order and how they go together. I started out in chromatography, especially gas chromatography. So I say I'm at the GC: adenine, thymine, guanine, and cytosine. So I'm at the GC, GC. When you talk about RNA, you're talking about the coding of life. This gets all the business done. So this is kind of like the business end of getting everything done. So you have the president of the company, DNA, and then you have all the minions, which is all the different RNA forms, and they are getting the business done of life. So here, instead of the thymine, you have the uracil. So you have the adenine and the uracil that pair together. Uracil is the pyrimidine and the adenine pairing together. And then you have the cytosine and the guanine, again, pairing together. Now, RNA is only a single strand, but because of that backbone, which is similar, it's not exactly the same. This is a ribose sugar and a phosphate instead of the deoxyribose and the phosphate. And you still have that twisting effect because when you start to stack them up, they do give that curve as well. So here's a little comparison of the form and function of DNA. Now, DNA has direction to it. You have a 3' end and a 5' end. That will become important when we start talking about transcription and replication. You also have some differences there's one strand for RNA. There are 2 strands for DNA. And your DNA backbone, like I said, is deoxyribose, but your RNA backbone is ribose. There is that change in the base where you've changed out that uracil for thymine. And the location of where these are contained within a cell or within an organism are slightly different. There are some exceptions, but for the most part, DNA is found in the nucleus, where RNA is found throughout the cell, in the nucleus, the cytoplasm, and it is a major component. RNA is a major component of ribosomes. DNA is the master code, and RNA is the coding, or the bits of coding for different functions. So DNA should not be changed. There shouldn't be alternate forms of DNA or changes to the DNA. The only time that it really should change is by mutation or by some sort of transcription error or aging or something like that. There really shouldn't be major changes to the DNA overall. And again, there are some caveats. But then RNA changes all the time. It changes, it picks up different portions of the DNA code. So it's constantly being recycled and reused. It's being changed over and over again. Now, if you look at a typical type of cell, you have your transcription going on in the DNA, and you have messenger RNA, mRNA, that is going to take the transcription of the particular set of codes from the DNA that is needed. And then when you need to make proteins or make other working chemicals in the body, biochemicals in the body, you have the translation, you have tRNA, the transfer RNA, bringing in amino acids. They're bringing in 3 at a time. They're bringing in amino acids, and then it's being assembled in the ribosomal or the rRNA, where it's being put together. And it kind of reminds me of Willy Wonka and the Chocolate Factory, where you kind of have this crazy machine taking all these colorful bits and pumping out a chain of candies at the other end. Well, in this case, it's a chain of a protein or some other functional groups. So why would we use DNA or RNA? Why would we want to be studying DNA and RNA? Well, there's all the obvious answers. We've all heard about crime scene analysis. With COVID, we've heard about the search for the code for COVID. You've also probably heard of genetically modified organisms like corn, maize, and other types of food. The agricultural component to DNA and RNA is quite large. And all of your sampling for a laboratory workflow for a general DNA and RNA is very similar. There's, of course, always some changes depending on exactly what you're trying to do, but the workflow itself is fairly uniform. You select a target. So let's say you are looking for the code of some of the crown proteins, some of the spike proteins on the coronavirus. You're looking for that particular code. That is your target, that particular sequence or code for that particular part of the organism. Then you choose the form of the sample that you're going to be testing. Is it a tissue? Is it saliva? Is it blood? Is it going to be corn? Is it going to be seeds? What is the actual, material that you're going to test? And if that material is a solid, like a tissue, like liver or heart, or if it's a plant material, you're going to have to lyse those cells and extract out genetic material. And most of the time, you're going to use some sort of mechanical or physical lysis method to kind of break open the structures. If you're doing like a plant material, plant cells are very rigid. They have a lot of structure. They have cellulose. They're very strong. And so you're going to want to lyse them or break them open and be able to extract out the genetic material. So then you're going to extract or isolate your genetic material. You're going to use lysis buffers. You're going to use different reagents. You might be using kits to isolate your genetic material. And then you're going to prepare it for processing and analysis. You might put it in a plate. You might use some other buffers or kits. You might use the primers or other nucleotides that you're going to be processing for. So you're going to prepare your sample. And then you're going to do whatever replication or testing process that you're continuing to do. And obviously, we're going to talk more about PCR, but there are lots of processes for replication and testing. So what is the source of your genetic material? Is it part of a virus? Is it a piece of tissue? Is it a specific cell? Is it a blood cell? Is it blood itself? And then what is the area that you are going to be working with? What sequence, what code are you going to be working with? So to do that, you take your sample, you use tubes, vials, you might use your homogenization like I showed, like a GenoGrinder. You put your grinding media in it, then you add whatever chemicals that you're going to use, your lysis buffers, your solvents, maybe some stabilizers or salts, and then you do your processing. And then we're going to get to PCR and the materials for PCR in a few minutes. So some people say, well, why homogenize? And why mechanically homogenize? That's a question we actually get quite a bit. Well, sample prep is one of the first important steps in doing any analytical laboratory process. This is where contamination can be introduced. It can be where if you make the mistake in the homogenization or in the sample preparation, you then amplify that mistake throughout your process. So you want your samples to be homogeneous. You want them to be easy to extract or digest or dissolve or whatever else you're going to do with them. And you want to be able to preserve whatever compounds or DNA or cell that you want without destroying it. So you're going to, if you're using a process that creates a lot of heat, maybe you're using a kind of like a coffee grinder or something like that generates a lot of heat, you might be actually breaking down some of your targets. So you might want a cooling feature. Another feature of why you want to homogenize is that the smaller the sample size, the less sample you need to ensure that you have homogeneous sample. So if you have a pencil eraser about 5 millimeters, a crayon, which is about 2 millimeters, a pencil, which is about 1 millimeter, and a fine point pen, which is about 5.5 millimeters, and you have a small amount of sample and you need a 1% uncertainty. So you really need that tight uncertainty. If you grind that sample down to a very fine powder, less than 0.5 millimeters, then you can ensure that you need less than 12 grams of that sample to ensure that you have a homogeneous sample. If you have a little higher uncertainty, and you can go up to about 10%, which is a lot of uncertainty for process, you could even get down to less than 0.1 grams. So then your sample amount becomes very, very small. So that also aids in your testing. At Specs, we have two premier pieces of equipment lines that do these jobs for you. If you have a soft sample, you're going to be looking for something like a freezer mill, which uses cryogenic grinding. Or if you have some, general laboratory samples and tissues and things like that, you can use our GenoGrinder, which is an all-purpose bead beater and vibratory mill. So these are two products, and I know we're going to get ready to demonstrate with Eric in the lab some of our lab preparation products. Joe?
Thank you, Patty. I'll take it from here. My name is Eric Smith. I'm the application scientist at Spec Sample Prep. Today, I'm going to give a brief overview of how to use the 2010 GenoGrinder and the PCR Max to improve your DNA, RNA extraction workflow. The 96 deep well titer plate will be used for the homogenization step. A small amount of sample is added to each well. After the sample is added to each well, we will add the lysis buffer to each well. After the sample and the lysis buffer have been added to each well, we now add one ball to each well using the ball dispenser. To simply drop one ball into each well, we press the lever and release. To seal the titer plate, we apply the cap mat by pressing each protrusion of the cap mat into the deep well titer plate until the titer plate is completely sealed from leakage or cross well contamination. After the titer plate is completely sealed, we place it inside the Geno grinder on top of a nesting tray. Here we have 6 titer plates with samples that will go simultaneously. We simply lower the lid with a quick release knob, give a few turns to lock the plates into place. We close the lid. After the samples have been loaded in the Geno grinder, you program the run, rate, 0 rest, 1 cycle, press run. After the run is complete, we lift the lid to remove the titer plates from the Geno grinder by simply unlocking the quick release knob. The titer plates are removed. From this point, we will isolate the DNA and purify the DNA during the cleanup steps. After the DNA has been purified during the cleanup step, we add the DNA mixture to the PCR tray with the PCR matrix and incubate. This concludes our presentation demonstrating the 2010 Geno grinder and the PCR Max. Now back to Leah Anderson-Smith to continue the sample preparation presentation.
All right, thank you, Eric. I hope you were all able to see that. Moving forward here. I'm having trouble advancing the slides. Joe, could you help me out, please? Thank you. So to capitalize or continue on with what Eric was talking about with sample preparation, I thought we'd talk a little bit more about why exactly it's important to have a good thorough homogenization of the sample tissue. This slide shows some samples that were homogenized with the Geno grinder. Down at the bottom, these were mammalian tissue samples homogenized with the Geno grinder. And you can see that they're very thoroughly homogenized. You have a colored, clear solution with no visible solid particles. Whereas in the 2 upper pictures, we have liver and spleen samples that were homogenized using a manual method with a handheld probe that vortexes the sample. And you can see in all of those samples that some of the tissue was not fully homogenized, leaving solid material, unhomogenized material behind. The sample liquid is slightly colored, but certainly not homogenized to the level that we see with the genogrinder samples. Carrying on from that, why this is further important is that the thorough homogenization results in a much faster sample preparation time. We found that through our studies that the pro-K digestion step is greatly reduced, sometimes from as much as 3 hours down to about 15 minutes, which means that the total preparation time for the sample is reduced from about 3.5 hours to about a half an hour, thus greatly enhancing the efficiency of the time spent in the laboratory. And in the graph over on the right, you can see that homogenization with the genogrinder also gives a much higher DNA concentration. This was measured by UV vis. And we compared samples here of mouse heart, chicken breast, and beef loin. And you can see in all three cases, the concentration of DNA obtained was much, much greater when the genogrinder was used to homogenize the sample. In addition, while the concentration was higher, we also obtained very high quality DNA. You can see in the agarose gels that were prepared and run by electrophoresis in the picture below, you can see that the bands for the samples prepared using the genogrinder are very dark, but also very discrete, which indicates that we've obtained not only high quantity of DNA, but also high quality DNA, which is very important for research. We've been doing some work also to prepare our own DNA isolation kits that are specifically tailored for use with the genogrinder homogenization method. And we've recently done some work to compare our DNA isolation kits with a couple of kits that are commercially available from leading manufacturers of DNA kits. And I think what's particularly important here is to highlight that all of these samples were prepared using the genogrinder for homogenization, but using the different DNA kits with the different buffers that they offer. And you can see that the samples prepared using the genogrinder and the SPECS kits give very high results that are comparable or in some cases better to the results obtained with the kits from competitors. Particularly if you look at the liver, spleen, and chicken breast samples from kit A, you can see that the results obtained with the SPECS kit were much higher. We're very excited that we're going to be offering DNA isolation kits for mammalian tissue in the coming weeks. And we're also working on developing DNA isolation kits for plant tissues, again, targeted for use with the genogrinder homogenization method. And at this point, I think I will pass this on to Alan.
All right, and I was able to change slides. Hello, everybody. Good morning, afternoon, evening, wherever you are. Good to be here with this group of people to discuss PCR. So we've now isolated pure DNA from our samples, and now it's time to talk a little bit about how we would actually use a PCR to analyze this material. I should point out that there are, and this is certainly not a complete list, but there are a number of other methods for analyzing DNA in a sample. Some people will use more cell culture type methods, growing a sample on an agar, a petri dish, and letting the sample grow. Obviously, the sample has to be alive for that to happen. PCR does not require that, so that's an advantage right there. There are immunological approaches like ELISA assays. If you have the appropriate antibodies, then you can work with that. That's helpful also, but you do not get anywhere near the specificity that you can get with certain PCR techniques. So we are going to focus on PCR. PCR, is all over the place. Everybody uses it. We hear it in the news, as Patty had mentioned earlier. With COVID, people are using PCR to analyze, either the whole virus or just the spike protein. Almost every area of science, people are concerned about pathogens and other organic materials as impurities in their products, and they need to be analyzed. A big example that's growing more and more lately is in the cannabis field. You definitely want to use a good homogenization technique. As Patty had said, plant samples are pretty hard to break open and lyse all the cellular material. A lot of cannabis labs use more manual techniques for homogenization, and they tend to use ELISA and cell culture, but recent data has shown that PCR is a much more accurate technique and certainly more appropriate for analyzing for pathogens that a lot of states are requiring. So exactly what is PCR? So PCR, of course, stands for polymerase chain reaction. It is a chain reaction. The products increase the speed of the next step, as we'll discuss. But it's also based on the science that Patty discussed at the beginning, and the whole issue of the genetic code and the central dogma about how everything that makes us human is stored in the chromosomes. Signals are sent to the chromosomes to create messenger RNA, which is then sent to the ribosome for creation of a new strand of that sequence and the creation of the corresponding proteins. So we're sort of simulating key parts of this step in the PCR reaction. So PCR is a temperature-driven process. When you mix everything together, and everything includes your sample DNA that you're trying to amplify, individual nucleotides, which are all the base pairs, sugars, and phosphate little pieces. The primers are selected ahead of time, and they have a sequence that matches the corresponding complementary strand inside the sample. And then there's a polymerase. We usually use a Taq polymerase, which is a protein found in thermophilic bacteria, and they can withstand the temperatures required to carry out this process. At the high temperature, the double helix of DNA is broken out into two separate strands. And then at the lower temperature, around 50 degrees, that's when the primers and the polymerase anneal themselves onto the strands. And then we warm it back up to around 70 degrees, and that's when the polymerase does its job and starts the elongation phase. So we plot this out on a temperature type graph over time. I'm showing here the first two cycles of a large number of cycles. And you can see at the high temperature, we have the denature step, and then the anneal step, and then the extension. That doubles the number of strands, and we go back to 95 degrees and denature the new double helices, and the whole process begins again, and it's done over and over again. This is a typical plot to show as the number of cycles increase, the number of pairs of DNA keep going up faster and faster. These graphs tend to reach a plateau phase at some point after a number of cycles, because you're starting to run out of nucleotides and you just can't make any more DNA. All right. There are a lot of different types of PCR. I'm only going to mention two main categories. One is the more conventional endpoint PCR. This is a qualitative method. This is really meant just to amplify whatever DNA is in your sample. Obviously, you have to be careful because you may end up amplifying an impurity, and that's why you want to make sure you have clean material. But like in forensics, they do not need much material. They just need to have a few molecules of DNA from the crime scene to amplify, and then they can analyze it. The way it's analyzed in endpoint is to run a gel electrophoresis. You can separate off the bands if you need to. But in real time, or qPCR, you actually add dyes to your process. And I'll show you the two general ways this is done in 1 second. But I did want to mention that there is some confusion, at least for me, confusion in the nomenclature of PCR. You'll see RT-PCR. That could actually mean real-time PCR, which is what I'm describing. It's a qPCR. But you can also do the same exact thing that we've been describing here with RNA. And in that case, you're using a reverse transcriptase protein. It's also called RT-PCR for reverse transcriptase PCR. All right. So this shows how this actually happens. And if you use a primer that has a fluorescence reporter and a fluorescence quencher on the same primer, this material will not fluoresce as is. The quencher is geometrically so close to the reporter that the fluorescence is quenched intramolecularly. This is called FRET, or fluorescence resonance energy transfer. And so there's no signal. But as the polymerase starts to work its way down the chain, it breaks apart the primer and releases the reporter. And every time this happens, you get a greater signal. And that's what ends up being the y-axis of that plot that I showed on the previous slide. The other general approach is to use DNA intercalators like CyberGreen, which binds into the groove of the DNA. And so the more DNA that's formed, the greater the signal. You can run multiplex qPCR by using a number of different fluorescence reporters, as long as they're within the range of the instrument. And Patty will have a little detail on that later, I believe. And in fact, Patty, I believe it's time for you to talk about instruments. Thank you very much.
Okay, thank you, Alan. Yes, so in our specs portfolio, we are very proud of our PCR Max products. And specifically what we talked about in this presentation, we have our real-time qPCR, our PCR Mac Eco, our real-time qPCR system. And then we also have our conventional thermocycler, our PCR Max Alpha Cycler. And we do have some more information in our downloads. You can download the different brochures with all the features, or you can contact one of our team directly and get some more information. One of the nice things about our units is that you are able to simultaneously conduct a number of different experiments because you can do gradients in our thermocyclers. So you can use the rows and the columns to test different temperature conditions and different reagents all at the same time. So you can really run that real-time experiment and see which particular combination of temperature and reagents are best for whatever target you're trying to do. So that is a very unique feature. And we also have calibrated a closed circuit camera and calibrated to the different emission spectra of many of the common PCR fluorescent dyes. So they're very easy to use. They can conform to any set of conditions that you need for your particular experiment, and they have easy to use software, and it has all of the features that we expect in today's instruments, where you can access by USB, and you can do all sorts of things and updates and things like that. So we are very proud to now be offering our PC Max under our Specs brand, and we look forward to helping you in any way possible in the future. So I think, Joe, I'm going to hand it back to you.
Yes, you are. Thank you so much, Patty, for that. And thanks everyone else for your contributions on the presentation today. I thought it was really informative. We've had quite a few questions coming in, but we always like to make sure that you have an opportunity to ask questions. So please do use the Q&A box and keep asking questions as we come into our Q&A segment now. So that's excellent. Right. Okay. Let's start with the first question. So where are we? Yeah. Okay. This one looks good. Right. Not sure who's going to answer this one, but I'm going to read it out. So what types of samples require cryogenic grinding instead of room temperature homogenization?
I can take that, Joe. Patty mentioned a little bit when she was talking that the Geno grinder is typically used for fresh sample tissue samples, such as could be used for plant tissue, leaf punches, can be used for seeds. It's also used for various mammalian or animal tissues, as we saw in some of the samples that I showed. But if you're dealing with tougher, harder to grind materials or materials that cannot be processed at room temperature, that's where the freezer mill comes in and is very handy. So this could be used often for samples such as bone, hair, teeth, fingernails, often used for samples that might be for forensic analysis or could be archaeological samples. Sometimes they're used for identification of samples from remains after an accident or a plane crash or something of that sort. So GenoGrinder is useful for most fresh samples, most tissue samples, but for those really tough samples like bone and teeth, that's where the GenoGrinder or where the freezer mill and the cryogenic grinding really comes in handy.
I could add in the the cannabis field. I was talking to a customer recently, and it's one thing to grind cannabis flower, but you ever try and grind a gummy bear down to a fine powder? Not an easy process, and you definitely need freezing for that.
Okay, brilliant. Excellent teamwork there on that answer. I love that. Another question is, what is the typical homogenization time on the GenoGrinder?
Most samples can be processed in a minute or less. Some samples, if they're tougher to process, require maybe up to 3 minutes. So typically it's between the 1 and 3 minute time. We found that in the work that was done here that we presented with the tissue samples that we used, some of the softer tissues or easier to process tissues, once 1 minute of homogenization was adequate, and in others 3 minutes. And in those cases where 3 minutes were used, what we did was it was processed for 1 minute with a 20-second pause and then an additional minute pause for 3 cycles.
Right, perfect. Thank you, Leah. Another question here. Can specs provide guidance on selecting the correct disruption tubes or grinding media and homogenization protocols?
And
sounds like another one for you.
The answer is,
Leah, you're popular today, Leah.
The answer is, of course, we can. We do have a document that provides some guidance on choosing the correct disruption tubes and grinding media for various tissues. I think maybe we're going to try to post that here, but certainly anyone could contact us and we'd be happy to provide that information. We are also happy to work with any of our customers on developing protocols that are suitable for the tissue samples that they need to process.
Okay, brilliant. Can you overcycle a sample? And if so, what happens?
I think that's something for Maddie there.
Maddie, you
Hello,
there?
can you hear me? Yes.
Hello, yes, we can hear you. Thanks, Maddie.
Sorry, can you repeat the question?
Yes, of course. Can you overcycle a sample? And if so, what happens?
Well, it depends what it means by overcycle a sample in terms of for how long. So we're going to need more details on that, because if you forget about the sample, then yeah, you're not going to get great results. But if it's like, I don't know, a few seconds, then it might be an issue. So it depends exactly.
Did you say 10 seconds is not an issue? Did you say?
Yeah, a few seconds is not an issue. I've done it. Probably quite a few of us have done it in the lab. And we had good results, but it depends exactly for how long you did it.
I mean,
Okay.
if the question is, can you run too many cycles? I mean, if you remember the amplification curves, as you get to larger number of cycles, you're going to have less efficient turnover because you're going to start running out of nucleotides. So I think part of it is whether you're doing quantitative PCR or qualitative PCR.
Yes. Usually if you do qPCR, you usually, I don't know, you finish around 35 cycles roughly. But it depends on the product you're using, of the equipment you're using. if it's a way you set up with our unit, yes, you can set up the number of cycles. Others on the market, I can't comment on that, but with our unit, yes, you can set up to run up to 35 or 40 if you want to, or many more. But also depends of the reagents you're using, and also of what you're trying to achieve, what you're looking to detect. Probably, like I said, if you're using a specific reagent to detect, I don't know, let's say, because with the current situation, COVID, yes, it's recommended you finish your cycles around 35, roughly, because after that, you're not going to have accurate results.
Okay. I think that's a very good answer. Thank you both for that one. So I had a question here. Do you offer primers as well, and can you customize primers?
Well, we do offer quite a number of primers, mostly a lot of different human pathogens, various diseases. I can't really address the custom primer design right now. Maddie, have we been doing any custom primer requests?
No, but we do offer detection kits for certain pathogens. They have everything that you need. You can purchase the kit with or without master mix, but basically the package contains everything you need to run a PCR. You're gonna need to do the extraction and with another kit, but for detection, we have our PCR Max detection kit, which offers everything.
Perfect. Thank you both. Which kind of precautions should be considered in using this DNA extraction machine?
I guess it depends on if they're asking for safety precautions. I think, Maddie, maybe you might know what kind of precautions.
Yeah, that's a good question, actually. I'm not sure exactly the question, if it's like the whole, like the procedure, the assay, what should we take in consideration, or just precautions. Yeah, I'm not sure I understand the question. So if you can follow up with more details, that would be great. And we can send the answer via an email.
Yeah, or maybe you can just resend the question to us. Just give us a little bit more detail around the types of precautions that you're thinking about there on that question. That'd be great. Thank you very much. Okay, have you ever been able to correlate the sample qPCR assays with the USP? There's a bracket 62 there. I'm not sure what that's referring to, but the pathogen identification. Can you determine the absence of specific pathogens?
I will take this one. So if you're using endpoint PCR, obviously the endpoint PCR is used to detect the presence of or absence of a pathogen. But if you use qPCR, then depends again of the kit you're using, and that kit will have the sequence for that specific pathogen. So, for example, if you're looking for salmonella, the kit will have the sequence for the salmonella and will tell you if it's there or not based on the CT values.
Right, okay, cool. This is a fairly interesting question, actually, that popped up earlier. Is PCR a good pandemic indicator? I think that's quite this is quite an open question, isn't it? I mean, there's probably quite a different ways you can answer that.
Yeah, I mean,
I think I will start with this.
Who wants to go first? Let's put it that way. Who wants to answer that first?
I will start with this. I think we've seen in the last year or so since the pandemic, we started hearing and seeing onto so many comments about qPCR. Is it the right product method to test for COVID? If we think backwards, like HIV and all of the other pathogens, we've been using qPCR to test. from my opinion, obviously everyone will have a different opinion on this, but from my opinion and experience, and also what we've been working with different researchers and hospitals, we do know that qPCR is a good method to detect pathogens, to detect COVID. And also, but keep in mind that qPCR is not only the product, it's not only the equipment, it's also the reagents and the sequence. And obviously, that's why in different countries at the moment, NGS, next generation sequence, is really important, because if there are some variations in the virus, everyone will upload the data in a database, so we can work if there are any, like I said, variations in the virus, and we can work to develop a new detection kits for the specific variations. But yeah, like I said, in my opinion, yes, and based on what we've seen and work with different knowledgeable people, yes, this is a good method to detect COVID. So if anyone wants to add on that.
The only other thing that I would add is there are a lot of specialized variations of PCR, and I'm certainly not experts in any of these, but the one that strikes me as very interesting is digital PCR, where, like, instead of having a 48 well plate of samples, let's say, so you're doing 48 different PCR reactions, each well is basically nebulized into tiny little drops, and you end up with thousands of drops per sample being analyzed. And if you're using multiplex, multiple dyes, it's supposed to be a very good way of quantifying, different variants that might be in there as well. Just don't know that much more about it, though. Something to look up.
I would like to just say that the question is it a good PCR PCR is a tool, and it's no more of an indicator of a pandemic than a gas chromatography is an indicator of pesticides. It's a tool to measure, or a tool to amplify the pathogen that is causing the pandemic. But I will say that without PCR, we would not be in the place we are now, which is distributing vaccines, because that would not have been probably would not have been done by now. The ability to really isolate and amplify and produce vaccines and all sorts of other things have depended almost exclusively on PCR to, and we have PCR to thank for the quick response and the quick ability to develop a vaccine. So it's not an indicator of a pandemic, but it has certainly played a very important role in it.
Oh, that's brilliant. That's a really good response. I really think that's brilliant. So there was a continuation from a question earlier regarding these pathogens. So do you supply a kit for the pathogen BCC, USP bracket 60 bracket closed? Have you ever been able to determine the absence of the BCC pathogen in a product using your qPCR? So I think that's a follow-up from an earlier question that the same person's asked, just a different pathogen.
Yeah, we'll have to have a look, because we have over 1,000 pathogens in our portfolio. So I will have to look for that specific one. And I will have a look at exactly the specification and the IFU for that particular pathogen. And I can send the information the next 24 hours.
Okay, perfect. That's great. Thank you very much, Maddie. Another question is quite long. I'm just going to I'll read it out as it is. So many modern qPCR protocols use only two-step denature, anneal, extend, denature, anneal, extend, versus traditional three-step, which is denature, anneal, extension. Will pre-amplification enhance performance, amplification efficiency, non-specific amplification in this type of protocol? I don't know if that made any sense to anyone, but hopefully.
Joe, I think you should handle that question.
We might be here a long time.
Sorry, when you just another follow-up on that, when you refer to this type of protocol, is the one that we discussed during the presentation with the genome grinder, or is it something else specific? But just as a general rule, if you don't follow the steps, the 3 steps, I don't know. Depends also of the essay and what you're trying to achieve. But sometimes it's working. I try that, and sometimes it's working to just jump on step. It's working, but again, it depends what you're trying to do. I will need more details on what you're referring to this type of protocol. So yeah, but like I said, it depends on the essay and what you're trying to achieve. But I've done it and it worked for me. So that's another thing to consider when you do a PCR. Optimization is key. So if you try something new, if you buy new primers, new reagents, I usually do optimization. And a few weeks ago, we had a webinar on optimization, and we provided some key, let's say, important steps with one of the professors that we are working on this. So yeah, sorry.
No, that's okay. Yeah, that was a really good webinar. Everyone should definitely check that one out on the on-demand platform. But the part of it says, will pre-amplification enhance performance? Is that what you're referring to, or is that something you can answer? Will pre-amplification enhance performance?
Like I said, it depends of what you're trying to achieve. But I've done that, and I think I've done it, if I remember correctly, I've done it based on a certain optimization, and I knew what I was trying to achieve. This was like a few years ago, so I don't have all the data. But I can have a look backwards and see exactly when I've done this and what data I've achieved, what information and the results I got from that. And I can share them.
Okay, that sounds really great. Thank you. Why did the WHO allow 45 cycles all through 2020? Is 45 cycles accurate to detect SARS-CoV-2? Or SARS-CoV-2, I should say.
I will take this one. So, World Health Organization is suggesting, based on what they are kits they are using, based on the reagents they are using. Bear in mind that when you do a diagnostic, when you follow a diagnostic, a CIVD, FDA-approved protocol, you follow what it says on the instructions. If on the instructions of the kit you purchase will say 35 cycles, then you do 35. Or if it says 40 or 45 cycles, then you're going to follow that one. You cannot change that. If you change that, then you are not going to follow the IFU. Therefore, that kit or the changes you've done, they are no longer according to FDA approvals or CIVD for Europe. So it depends, but I would say for anything that is diagnostic related, follow the IFUs, because those ones have been optimized, checked, and these are the ones that we should follow. Obviously, the World Health Organization will suggest different things, but that should be taken in consideration based on the reagents that are used, and should be taken in consideration also the manufacturer of the kit, and not the person in the lab that does the test, because at the end of the day, we have to follow the IFU. I think I've answered that question, but if it's more on that, please follow up with further questions.
Yeah, no, that's great. Thank you very much, Maddie. Someone else has asked, have you had any experience with this grinder, the genome grinder, with the algae samples? So specifically, cyanobacteria, blue-green algae. I think I pronounced that correctly. Probably didn't, but Sounds like one for Leah, possibly.
Right. I don't know the answer specifically. I know we have worked with a variety of samples, and I'm sure we have done algae samples. I don't know that we've worked specifically with cyanobacteria